Carsten, thank you very much for, let's say, inviting me to give this webinar. Actually, it's my first one, so we'll see how it's going, talking to a computer screen instead of being in a lecture hall. I hope you will enjoy. So the topic I would like to focus on is open data, linked open data, and how we exploit them in ligand-transporter interaction profiles. So it will be about data coming from different types of domains, could be different domains. For example, as you see here, could be the data out of our group, could be the data of ChemBio, a public depository, could be the data of a pharma company, just having one logo here. I apologize for company representatives being present from other companies, and maybe integrated data like they are in Open PHACTS, which means data covering different domains. But before explaining a bit more in detail what Open PHACTS means, maybe also let me explain the use case, drug transporter. So since 1976, we know P-glycoprotein. You see here the X-ray structure of mouse P-glycoprotein, and what it is doing is it's just sensing toxic compounds when they are diffusing through the membrane, and by use of ATP, pumping them out of cells. And how was it discovered? It was discovered as being responsible for multiple drug resistance in tumor therapy. That's basically obvious. So you have a tumor cell. The cell is just targeted by toxic agents, so classical anti-cancer agents, natural product toxins. And the cell is realizing there's something toxic coming in. It's upregulating this defense mechanism, and P-glycoprotein is just there pumping the compounds out, which actually means in therapy you get resistance. We know this protein now since 40 years. We know that we can block it with a lot of compounds. We know its role at the blood-brain barrier. We know its role in ADMET, at the intestine, at hepatocytes, blood-brain barrier, at kidney cells. So you see you have a lot of transporters. It's not just P-glycoprotein. We have 48 so-called ABC, so ATP binding proteins in our human genome. And there are other transporters too, which play a role. We have more than 800 transporters in our genome. And it's quite interesting to see the interplay, to learn more and more how they influence bioavailability, how they influence elimination, distribution, and also toxicity. And later on I will present to you a use case, how we include, for example, transporter models into type of in vivo predicting systems for predicting in vivo toxicity. So 5 years ago, when we started the Open PHACTS project, how looked our workflow like when we wanted to make computational models, for example, predicting whether a compound is an ABC transporter or not. So here we are talking about the breast cancer resistance protein, and what Florian, just yesterday she had her defense as a PhD student, what Florian did is she spent several months in compiling data from literature, which actually means not only looking for the different publications, but taking them, extracting the compounds out of SAR tables, redrawing them, putting them in a database, and then doing machine learning. And for example, what you see here is a decision tree model for BCRP inhibitors. So using MOE descriptors. And finally, yeah, we got quite a nice model. Area under the curve is around 0.8. And the training set here comprised around 700 to 800 compounds. And we did, of course, we did the same for BSEP, we did the same for OATP1B1, OATP1B3. But it's quite time consuming, taking several months going into each individual paper. You need to decide, are the assays analogous, how to, let's say, set the threshold for active, inactive. And that was also the time when finally the Open PHACTS project started. But I guess before explaining to you what Open PHACTS actually means, there will be a poll done by Carsten.
Yes, we have prepared two polls for you. We want to know you, get to know you a little bit better, see what kind of scientists we have in the audience. And the first poll is about what's your background? So you have a few choices here. Would you consider yourself either a medicinal chemist, a computational chemist, crystallographer, biologist, or any other kind of science? And the poll will run for a few more seconds. You can see there's a more or less clear winner. You give your vote now, and I will close the poll in 5, 4, 3, 2, 1 now. So we do have about 30% medicinal chemists, 45% computational chemists, no crystallographers, a few biologists, and the rest is other. Thank you very much, and I will hand over back to Gerhard.
Okay, so it's medicinal chemists and computational chemists, maybe also computer scientists. That was also the quite interesting mix we had in Open PHACTS. So what is Open PHACTS about? Open PHACTS is an IMI project. That means Innovative Medicines Initiative. It's a public-private partnership of large pharmaceutical companies, so they need to be FPA members. I guess we have around 40 or 50 FPA members in Europe, need to be Europe-based, and between the European Commission. IMI 1 was 2 billion euro, one coming from the pharma companies as in-kind contribution, the other billion euro was coming from the European Commission. IMI 2 actually is, I guess, 3.2 or 3.3 billion euros, so even larger. What is it about? It is basically bringing academia and industry together, doing research on pre-competitive needs for pharmaceutical companies. So needs where they can work together without infringing IP. So one example could be kinetics. I guess every or a lot of companies are interested in, can we translate off kinetics or residence time into in vivo efficacy? What would be concepts there? That's K40D, that's one IMI project running. Another possibility would be, can we predict the outcome of the 28-day rat tox study? Can we do computational models where we want to predict this? I will later on showcase one of these examples, saving animal experiments and being just faster. So the slogan of IMI 1 was new safe medicines faster. This also, every company has the same demand here, and you can think on use cases where basically there is no competition. And Open PHACTS, another IMI project, is, can we link public data sources to our in-house data sources? Can we mine them? Can we link them? Because this is also something every pharmaceutical company finally has to do. They have to match their internal data warehouses with what's out in the public domain. So when we started, the first question was, okay, which data sources, which public data sources should we link? There are more than 1,400 life science data sources in the public domain. The question is which ones to take. The approach we took is, let's just make a survey among our pharmaceutical partners. So Open PHACTS finally stands for the Open Pharmacological Concepts Triple Store, comprised 30 partners, ran for 5 years. It just finished 2 months ago, was around 20 million euros, just to give you the figures behind. So the first task was, let's make a survey, 80 business questions, like what is the selectivity profile of known P38 inhibitors, or let me compare physicochemical properties for known oxidoreductase inhibitors, find me compounds that inhibits targets in a certain pathway, acid only in functional assays. So this type of business questions came back. We collected them, sent them back to the companies and asked for ranking them. The top winning business question was, give me all oxidoreductase inhibitors with activity in human and mice better than 100 nanomolar. And actually, to be honest, I thought that's quite trivial. When we tried to solve it, it was by far not trivial. It took 2 postdocs 3 days, and here you already see the value. It's a lot of resources which need to be spent in every single pharmaceutical company to do this more or less on a daily basis. Map public sources to internal data warehouses, or just link them together. So this type of top-ranked business question already prompted us towards data sources which are very high on our list to make them interoperable, to connect them. One of course is ChemBio. It's the, let's say, compound target annotation pharmacology data source. Drug bank, where all the drugs are in. Wiki pathways, where are the pathways? We have then now DisGenet for diseases. So we started to link, identify public data sources with respect to compounds, targets, pathways, and diseases, and building up the platform. So how is it looking like? The platform basically, here you see, it's composed of the data sources. Data sources are provided semantically encoded. That means at RDF triples. So each data source, for each data source we interconnect. We get them in an RDF format, and then we interconnect, let's say, the RDF versions of the data sources, and on the fly, it's basically via the semantic triples, it's connected, and with a structured API, with structured calls, you have the communication to the user. So this is basically what is done in the Open PHACTS Discovery Platform. Here we get the data sources regularly for updating and for loading them in, and we developed the type of a basic GUI. We did not put a lot of effort. We basically took it from Lundbeck, so that was an in-kind contribution from Lundbeck, and adapted it to the Open PHACTS API, where you can do some basic queries. We developed standards. Of course, you need to develop standards. For example, a standard for RDF triples, suitable for the Open PHACTS Discovery Platform, so clear rules how the RDF version should look like. And what we also did in the course of the project, and I will present one quite successful application, we showcased applications. So what can you do with a type of large triple store, where you have data sources like ChemBio, DisGenet, Wiki Pathways, GeneGo integrated? What can you do with this type of data? Of course, we also had challenges. One was units. So when we looked for this better than 100 nanomolar, why did it take 3 days? One reason was because there's not one oxidoreductase, it's a whole enzyme family, so you need to look them up individually and species selective. And another problem was that at that time, we saw in ChemBio more than 5,000 types of activity, of ways how to encode biological activity. And doing a type of a manual assessment, that's time consuming. Now, and there you see the advantage of a type of a multi-collaborative effort. EBI picked it up, and now we have pChemBio values, which makes it much, much easier to compare, for example, biological results and data. So this was one of the challenges, the let's say, more or less unorganized way how biological activities is described in data sources. Another challenge was legal issues. After a few months, we knew we can do it. Took us almost 1 year to get, let's say, certainty that we are allowed to do it. So at the end of the day, we hired John Wilbanks from Creative Commons, who made us a license model, which allows basically to, on top of the platform, to exploit the results for pharmaceutical research and also for commercial exploitation. This was solved in a way that the RDF version we get under a certain license, which allows finally Open PHACTS users to use the results also for example, making cool applications, software for applications. One is the Open PHACTS Explorer, so that's just the web GUI. By the way, it's completely free. You can just go there, www.openfacts.org. You go there, you start playing around. Would be nice if you register, because then we know a bit better our user community. We don't track searches. Of course, we don't track that because it's pharmaceutical companies also using it. So we just know that from certain countries, institutions, who is accessing the platform, but there's of course not at all any track of the searches. So you can go there, you can just start using it. If you would like to develop your own application, also that is free. You register as a developer, and then you get an API code and can start playing around with the API. So this is how the standard web interface currently looks like. And here you just see a search I did, a search for multi-drug resistance protein 1, P-glycoprotein. And here you see, okay, you have more than 5,000 results. What's important, I'm very often asked about data curation. It's very important to notice we do not curate, we just integrate. We integrate the data sources as they are. What you can do as a user, you can show or hide provenance, so you know where the data are coming from. If you say show provenance, you get a clear indication. Besides each data point, you get a sign where it is coming from, so it's up to you to say, okay, I trust this or I trust this not. The real power of OpenFacts is in the API. The API allows you to build applications. Here you just see a type of a logo cloud of applications which have been built either during the lifetime of the project as example applications like ChemBioNavigator or PharmaTrack or the target dossier, or you see external applications where people, SMEs, picked up already the system and made on top of it like cool apps. For example, you have here, I guess, I don't know whether it's Android or iPhone app where you can search the OpenFacts platform via a mobile device. I would just focus on two applications. One is integrating it in MOE from the chemical computing group. So MOE offers the advantage to have SVL as a programming language, so it's very, very flexible, so that allows you just to integrate it, for example, connect it to the Open PHACTS Discovery Platform, and you basically can run a search and then directly import it into an MDB file, and you can start doing your QSAR stuff. What I would like to showcase in a bit more detail is the ChemBioNavigator developed by Biosolve IT. So this is very nice workflow, for example, for connecting already data extracted from the public domain, from OpenFacts, for example, with in-house data sets. So what is the ChemBioNavigator doing? It allows you to visualize the chemical and the biological space of different molecules. I just have a few screenshots, briefly, quickly guide you through what this tool can do. With the latest upgrade of the OpenFacts Discovery Platform, currently, I guess it's not updated to the newest API, but it basically showcases what you can do when exploiting the API. So you just type in, for example, a molecule name or a target or a SMILES code or whatever. In that case, it was sildenafil, so Ecker. You get the structure. Yellow means the yellow spot here means it's in OpenFacts, so there are data available. You can then, on the left side, you get the full compound profile, you get the SMILES, you get the INCHI, you get the INCHI key. By the way, if you take the INCHI key and paste it into Google, you make Google to a chemistry search engine. So just try that out. Take the INCHI key, paste it into Google, and you will find everything about sildenafil, or not everything, but a lot of information. You get some physicochemical descriptors. So here you see the provenance, where they are coming from, like for example P from ChemSpider, PSA from ChemSpider. You get target profile, everything that's data coming from databases. In this case, mainly from ChemBio. And at the end of the day, you can also run a similarity search. So for example, you run a similarity search, just define a similarity threshold and the similarity matrix, and you find analogous compounds. And you see, for example, You can plot molecular weight against van der Waals volume, so the system allows you to do several types of plots. The latest version also included PIC50 values, for example. And if it's filling up, you know there are biological data in Open PHACTS present for these individual molecules. You can show them in a type of a grid and single view, so that you can just hop across the different compounds. You immediately get a feeling about the sites of variation. You get a feeling about how does these SAR series look like, and what, for example, you also could do if you don't want to do a similarity search. You can do a substructure search, and with the substructure search, for example, I really like this tool because you just click on the bonds and it's highlighted in green, and once you are ready, this is the substructure you are searching for. So this is, and then again you get this type of new compounds retrieved, and you again can start analyzing them. So this just showcases how a system like Open PHACTS with a defined API can be exploited for making certain applications on top of the system, on top of the API. After a few years, we realized the power of workflows. So now you have a full suite of KNIME and pipeline pilot nodes available, and you can include Open PHACTS searches into your KNIME or pipeline pilot workflows. Here you just see a workflow we recently set up. We have one for P-glycoprotein, breast cancer resistance protein inhibitor profiling. This already has been published. This one, what I'm presenting now, is basically unpublished stuff. So this is a KNIME workflow where we basically do the following. Sorry for this a bit crowded slide. I just guide you through the main tasks. So we search for SERT and DAT inhibitors, SERT and/or DAT inhibitors. We retrieve all the compounds. You see here it's 9,300 bioactivity reports in the Open PHACTS Discovery Platform. We filter for IC50 to have a type of comparable activity values. Then you see here for the two targets, you have different amount of compounds, and then we just keep only those where we have measurements on both SERT and DAT. And then we put them forward, cluster them into scaffolds. We get interestingly 800 scaffolds, but I will focus on that soon. And then we start analyzing the scaffolds, and at the end of the day, we end up with a set of scaffolds where we have enriched compounds having higher activity at DAT than at SERT, and a set of scaffolds which are more SERT selective. So you can start exploring, let's say, the publicly available compound space for privileged scaffolds for certain targets. So what you see here are some of the scaffolds we identified. When looking on them, okay, what you realize is you have by far less DAT selective scaffolds than SERT selective scaffolds. Okay, this is also very important to note. Finally, it's extracted out of an integrated life science data platform, which is basically filled by publications. So Campbell is based on publications. And those of you who are in CNS, I guess they know that SERT is, let's say, the standard target, or at least was the standard target for antidepressants, while for DAT inhibitors, there are not so many, let's say, broad community applications like depression. So naturally, you will find a higher diversity for SERT scaffolds than for DAT scaffolds. What's also important to note when we analyzed in more detail the DAT scaffolds, I was wondering, why don't we find more cathinones? Because cathinones are basically illicit drugs, so there should be a lot of them in literature. And finally, when looking on them, you maybe see why. This is the way how the clustering works. The clustering works basis of rings. the scaffold-based and the scaffold algorithm. So this scaffold is different from this, is different from this, it's different from this. With a medicinal chemist-based view, you immediately realize that's definitely belonging to the same group of compounds. So this is something when I'm giving courses, when I'm talking to students, I always say, okay, using computer technology doesn't prevent you from keeping your eyes and your brain on, because with a medicinal chemistry point of view, you see it. It's a bit more difficult to set filters in a way that finally all these compounds pop up in the same group. Nevertheless, finally we did it. We compiled the sets and we retrieved a data set with 56 compounds, and what we noticed is basically it seems that the substituent here in alpha position is somehow triggering selectivity. We did, I would not say it's a QSAR, we just did a multiple linear regression analysis, checking if we see a signal, and indeed finally we have, when we look on the dopamine transporter PRC50, the size of the substituent here statistically significantly plays a role, and it also plays a role for selectivity, because basically it doesn't really play a role when it comes to SERT inhibition. So going one step further, we docked these compounds into the serotonin and the dopamine transport, and this is also something I would like to emphasize. Docking is nice. Docking gives you nice pictures, but you need to be aware when you do docking, this is the reality. This is just imipramine docked into the serotonin transporter. These are the top 100 ranked poses, and basically all of them are plausible. And to come to pictures like this, you either focus on a scoring function, which is quite risky, or you validate your docking poses by experiments. And this is what we are normally doing. And there I would just briefly like to explain our common scaffold-based clustering workflow. So here you see a use case for P-glycoprotein, so one of these ABC transporters. What's the challenge here? The challenge here is the polyspecificity of these transporters. So we have basically a binding zone of 8,000 cubic angstrom. So it's 10 times larger than a normal drug binding site is looking like. So when you dock compounds here, you find hundreds of poses, and definitely you find them more or less everywhere. And the question is how to finally come to a consistent docking pose. What we do in this case is, and this is why we are keen on SAR series. So when you start extracting from literature using this workflow, taking certain DAT compounds, nailing them down to a set of scaffolds, taking one scaffold, going back and see, okay, we have 56 compounds where we have data on SERT and DAT. This is exactly a data set then we can use for, let's say, guiding our structure-based approaches. So what we are doing here, we dock a selected set of compounds where we have clear SAR. So these are proper phenotype inhibitors of P-glycoprotein. We know we don't need this OH group. So there's basically just, when we scale according to lipophilicity, no difference. We need this carbonyl group because it's acting as an H-bond acceptor. And every time when we introduce a hydroxyphenylpiperidine here, we gain 1 order of magnitude when we compare, for example, with compounds like this. We dock these 5 compounds individually, and then we only analyze those type of poses or pose clusters where we have all 5 compounds in and where we have an overlay of the common scaffold. So this is the common scaffold. So we make a common scaffold clustering and we look on those poses, on those clusters where we have all the 5 compounds in, and see if these clusters allow us to explain what we know. And here finally, this is what we end up with. We see the H-bond to this hydroxy group here, from this tyrosine. We see another hydrogen bond from this tyrosine to this carbonyl group. So that's a very nice binding hypothesis. Actually, we published it in PLOS Computational Biology. We did The same for imipramine and the serotonin transporter, and we also in Nature Chemical Biology did the same for the azepam analogs and the GABA receptor. So finally, that's quite a nice approach. How can we validate our process? We validate them twofold. One is using ligand scout and extracting a pharmacophore model, a structure-based pharmacophore model out of the pose, screen a vendor library, order the top-ranked compound, test them. This worked very nicely with this approach. We identified 2 new PGP inhibitors with this approach. Another possibility, of course, would be doing mutations. In that case, that failed. Why did it fail? Because it would be obvious, you know, having phenylalanine here instead of tyrosine, you would expect a drop in affinity. Actually, it did not work. It did not work because P-glycoprotein is a symmetric protein, more or less symmetric, pseudosymmetric. So the ligand can enter from 2 sides, from the front here, from the back side. On the front, you have 2 tyrosines. On the back, you have 6 tyrosines. The template is a bit asymmetric, so we had only binding poses where we had the 2 tyrosines. But of course, the tyrosines on the other side can take over. So mutating these 2 doesn't really finally matter. But what stays are the hydrogen bonds because they were type of predicted in the pharmacophore-based approach. So when we did now the same approach for docking these cathinones into the dopamine and the serotonin transporter, the picture was not, finally was not that clear. We saw that we have type of 2 potential preferred binding modes. It's quite small molecules. What we definitely saw is when we have large substituents in alpha position, we see a type of steric hindrance. So basically the docking supports what we see in the SAR and what we see a type in the multiple linear regression analysis. We also see that in the serotonin transporter we have a type of lower scoring values, and we see that in dopamine we have hydrogen bonds we don't have in the serotonin transporter. So basically it comes a bit in a way together with what we see in the structure activity relationship studies. We also used SeeSAR. SeeSAR is a nice tool to finally get deeper knowledge on especially also solvation, desolvation factors when you look on binding modes. And what I like very much here is this type of visualization. So that means, okay, maybe I need to look a bit stronger here. That's nice, that's fine. Here maybe I have some problems, I have a penalty. When you look on another pose, you see, for example, here, seems that one of these 2 substituents maybe has here type of a clash or a penalty or a solvation or desolvation penalty. So what SeeSAR allows us is now to look on the individual poses in a type of an almost orthogonal approach and analyze them with a special focus on which part of the molecule maybe is favorable or unfavorable, contributes to the overall delta G, and you also get indications what could be the reason for this favorable or unfavorable contribution. We got here additional hints. That's very, very recent result, and I guess offline I will then talk with Carsten how we could interpret them, because currently, to be honest, I don't really know how to interpret it, but in other use cases, in other examples, it really guided our way how we could, for example, either optimize a molecule or at least get another hypothesis why, for example, this substituent is good or this substituent is bad. And I guess now it's time for having another call.
Yeah, this is a perfect time for the second poll. I don't know how many of you guys have had some experience with SeeSAR already, but this question could be with respect to almost any medicinal chemistry kind of software, but the question is in particular with respect to SeeSAR. So obviously we are always looking at enhancing SeeSAR, and we are asking you what kind of new features you would like to see in SeeSAR that you would embrace. This could be multiple proteins loading into the viewer. Currently, you can load one, then transparent surface for getting a better overview of the binding site and the ligand together with the surface, so the surface is not blocking the view. a pattern checker on the fly, a synthesizability checker to check if the edits that you have made in SeeSAR are plausible from a synthesizable point of view, or maybe expanding the editor to have more elaborate editing functions, such as a full list of functional groups, for example, and the like. And I see that the votes are coming in. I would wait a little longer, and I will close the poll in 5, 4, 3, 2, 1 now. Okay, so it's a pretty even vote here. A little bit emphasis on the multiple protein loading and the synthesizability. The editing functions seem to be fine, at least for those of you who know SeeSAR. And then there's also some emphasis on the pattern checker. Okay, thank you very much for that. And I will give the mic over back to Professor Ecker.
Okay, but
yeah, I think you got kicked out of the presentation.
Okay. Since we work again, at least I am now back in presentation mode.
Good. Yeah,
so
we can see that. Good.
If I can add, I would also favor the multiple protein loading because this that would be. So if I have as a presenter, maybe if I have 10 votes, we have a clear. Okay, so at let's say the remaining minutes, I would like to go now add another layer of complexity and talk a bit about our approaches, how we exploit, for example, data from Open PHACTS for doing in vitro to in vivo translation. So when we would like to predict toxicity, basically we have two possibilities, or three. One is we do the mechanistic approach. So we predict the effect of a compound on a target. We know how the target interacts in a pathway, and we know how this pathway influences the disease. So for example, it would be, if we predict BCRP inhibition, we should get an indication for cholestasis, because BCRP is transporting bile acids, so BCRP inhibitors more or less are prone to cholestasis. Another possibility is you just take the data as they are, do machine learning, and try to map the compound matrix to the in vivo effect. So that's classical machine learning. You take actives, inactives, and make a model. What we try to do is somehow to mix it. So here you see one approach we published 2 years ago. These are 40 antidepressant drugs, 20 targets in the brain, and this is the interaction matrix. So red means highly active. Here you see the PIC50 values. Yellow means intermediate, green means inactive. So what we did here, we extracted this matrix out from literature, and then in an OPLS correlated it with side effects extracted from clinical meta-studies. When you see a correlation with OPLS, so when you see there is somehow a link between this interaction matrix and a side effect, it did not work with efficacy, interestingly, but antidepressants, those who are in CNS, you know, it's quite complicated. Some compounds have a rapid onset, a slow onset, how to, let's say, quantify the degree of depression. That's not that trivial. So finally, we could go forward. We couldn't really go forward here. What we could go forward is side effects. When you get a model, you can trace it back. And the cool thing is when you do OPLS, that you get the individual contribution of each of your variables. So you see the interaction profile, which leads to the side effect. And there we could somehow extract out of the data a link between 5-HT6 inhibition and weight gain. Another use case is hyperbilirubinemia. This was done by Eleni in my group. So here you just see background of hyperbilirubinemia. A lot of transporters are involved in bilirubin level. We focused on OATP1B1 and OATP1B3. So what we did first is making OATP1B1, OATP1B3 inhibitor prediction models. So using different set of descriptors, using different classification algorithms, finally having a type of a suite of models for both transporters, where we, when we do predictions to a type of a consensus voting, how did we validate our models? We just screened drug bank, identified compounds which are flagged as OATP1, OATP3 inhibitors, but there was no indication in the literature for them, ordered them and tested them, and you see in 8 out of 10 cases we got quite a decent OATP inhibition activity. And then we used these models and combined them with, let's say, normal descriptors. So what we are using here is we extract the data set for compounds causing hyperbilirubinemia. Here 86 positives, 750 negatives. We characterize the compounds by chemical descriptors plus their transporter inhibition profiles, their predicted transporter inhibition profiles. So we mix somehow the computational, let's say, machine learning approach with the mechanistic approach by including predicted transporter interaction values as descriptors. And finally, when you also here do a classification model using RT-KIT for the descriptors, you see we get sort of the signal, not very strong, but basically we can maybe predict hyperbilirubinemia, and what's also important, OATP inhibition basically matters. So this stays as important descriptor in our data metrics. At the end, maybe one additional application for exploiting open facts. So that was just published 2 weeks ago in medchem.com. It's how to use it for phenotypic screening. So you can take your compounds from a phenotypic screen. You can start exploring the target profiles, the pathway profiles for these targets, the disease profiles for these targets, and start analyzing this data. So basically there is no longer a real limit. When you look on possible exploitation, so give me all pathways related to the regulation of P-glycoprotein and all the compounds hitting the targets in these pathways. Or my favorite business question, give me all compounds annotated with liver toxicity and the interaction profiles with all transporters expressed in the liver. Okay, we still miss maybe this part because that would mean text mining, so the first part. But still, these type of business questions, use cases become possible now. You don't need to take care of integration. That's done by the platform. We have already platforms for doing KNIME workflows, for doing repurposing, for doing toxicology read across, and very importantly, the most recent release of Open Facts includes also a full suite of bioannotated patents. So SureChemBio, you know, SureChemBio is now in Open Facts with bioannotations and fully searchable using our API. So I guess the limitations are really no longer in the computer. They are more in how you put together a workflow to maybe get your business questions answered. Of course, always be careful on the quality of data. We are not curating them. That's one of the key messages also I would like to give. How could you get engaged if you would like to be closer to Open Facts? The EU project ended, so February 28, finally it ended. End of April, we delivered our final report. So to avoid that this system is going offline, as a lot of systems which have been funded by public money, you know how this goes. You have the funding, you build up a cool tool, funding is gone, 2 years later the tool is gone, or maybe 1 year later the tool is gone. We had a strong emphasis on sustainability. So we founded a not-for-profit organization located in UK, a charity, the Open Facts Foundation. So the Open Facts Foundation is membership-based. So you become a member, you pay a membership fee, and in that case, and if you are a member, then you can already participate in driving the next developments. Currently, it's Lilly, GSK, and Janssen, and I guess also Novartis, but I'm not sure. So and these companies are currently members. Also, the University of Vienna is members. Also, for universities, there is the possibility to become members. Of course, here payment is different. Our payment is more or less type of in-kind contributions in the development or this type of services. So if you're interested, just go to www.openfactsfoundation.org, contact us, and we can start getting in more concrete negotiations with you. So that's finally the end of this webinar. I would just like to thank my group. Basically, they are doing all the work. I'm just sitting here in front of my computer and presenting it for you. Thank you very much for joining.
Thank you very much, Gerhard, for this wonderful presentation. I see that there haven't been any questions so far, so I guess it was a very clear and concise talk. Towards our audience, if you guys have questions towards Professor Ecker, then I ask you to please enter those in the questions box in the panel, in the GoToWebinar panel, and we can then address them. I'll give you a few more minutes to do that. And maybe a question towards you. When was the project again started? How long has it been around now? It's been quite a while, hasn't it?
Yeah, it was 5 years. It was originally a 3-year project.
Yeah.
And it was extended by 2 years, so it was 5 years.
So it must have been quite successful because of the extension then.
Yeah. It was, I guess, normally my projects are 5 years. When we started it and I saw it just 3 years, I asked our colleagues from industry, because they define the boundaries, why they had only 3 years. And they said, yeah, because we need to start fast to get it really running. So there was no 1 year of storming, forming, performing, team building. We more or less, we did the survey, for example, for the business questions even before we started.
Okay.
So we had already ranked business questions in place when we had the first consortium meeting. So that was and then after half a year we had a prototype. Our computer scientists, they named it Gerhard 0.1. That was quite interesting. Right. And then they told me, okay, now we throw it away. I said, why the hell you throw it away? And they told me, okay, because doing it was just for team building exercises among the programmers. Now we do the real stuff. So I learned a lot in this interface of medicinal chemists. I consider myself being a medicinal chemist, although I have this full professorship in pharmacoinformatics, and computer scientists doing coding. It's very, very interesting to see these different worlds coming together, doing hackathons. So if the computer scientists have a problem, they just rent a house somewhere in the middle of nowhere, order pizza and cola, and then sit together for 2 days and just start typing code. Medicinal chemists maybe do it completely different. So we had this type of hackathons. We had user-thons, 1 user and 1 coder, 1 medicinal chemist, 1 programmer sitting side by side. I said, oh, it would be cool to have this. Steve Pettifer said, okay, that's doable. I said, and this one? He said, no, come on. If we would need to do this, we need to start from scratch. So that was very, very helpful. The computer scientists learned our need, and we got the feeling this is easy to implement and this is not easy to implement. And then we had researchathons. So that was the phenotypic screening was one. Just scientists coming together. What would be a potential application, what we are aiming for? And then we identified analyzing phenotypic screening results. What do we need? We do need this data source, this API call, this workflow. That finally ended up in the MedChem.com paper.
Yeah, and it's always interesting to understand the use case behind something that you want to implement. So I guess that's a good combination, having a computer scientist and then a medicinal chemist who has to really describe the workflow that they want to accomplish, rather than wishing for a feature. Because just wishing for a feature is usually it's easy for the programmer to grasp this, but it's much better to understand the full usage behind a feature, why this this feature has to be in existence. This is also a concept that we use a lot when we further develop SeeSAR, that we go from use cases rather than from features. Yeah, and one other question I have, you said that you don't track the usage. Obviously, that's important for pharma companies. You mentioned that, but did you get any feedback from colleagues in the industry if they are actually using that, the interfaces, the data?
Yeah, we see, for example, especially those companies who are also members in the Open PHACTS Foundation, they use it a lot. We have one of our most frequent, we have several million hits currently per week. So one of our most frequent, let's say, customers is the SME doing all the library assessments for the lead factory. That's another RMI project. So they are doing that via the Open PHACTS platform. So that's a lot of hits for every compound doing this type of similarity search and then looking for the targets and pathways, the diseases. So yeah, we see that it's quite a mix of SMEs using it. So those already having applications on top of the platform. We get queries from there, and we get a lot of queries from pharmaceutical companies, especially those who are members in the foundation, because they all trust this type. GSK did a security check, so the system is considered there's a secure line, it's a trusted hoster, queries are not stored. Some pharma companies trust in a way that they send queries across the internet, others do not. This is why we develop currently a virtual machine that was the demand of Lilly, and it's already the beta version tested by Lilly. So there will soon be a virtual machine available, so you can have it fully in-house.
Okay, well, there are no more questions other than one question, which is asking if there was a video of the webinar available. That is indeed the case. We have recorded the whole webinar, and that will be available on the biosolvit/webinars webpage in a few days. So keep checking back. Also, the June webinar will be open for registration in mid-June. So always watch this space for new things happening, and we always put new webinars up there way ahead of time, so you guys are informed. So let me thank Professor Ecker again for a wonderful presentation. Thanks to the audience for taking time to watch this, and then we will see you back in June. Thanks very much.
Okay, bye-bye.